Good afternoon to all of you. Now you will be listening to Dr. Amitabh Mohanty, who is currently Program Director, National Genomics and Genotyping Facility of DBT at NIPGR. Amitabh Mohanty, he did his PhD from University of Delhi. He completed his PhD in 2000. And then he worked there as visiting scientist in the rice genome sequencing project, which you all might be knowing. So for that, he initially went for training to Tsukuba, Japan, where he learned some of the technologies, and he came back. He trained some of the people who are working in the rice genome project there in University of Delhi South Campus. And then later he moved to USA for his postdoctoral work. He did postdoctoral. He was worked as postdoctoral fellow at University of Massachusetts Medical School from 2000 in 2002, and then later he moved as postdoctoral fellow in Cold Spring Harbor Laboratory. From there, he worked in 2002 to 2007. Almost about 5 years he worked there. And then later he joined the industry, DuPont Pioneer Industry, and then later which is renamed or merged into Cartiva. So there he worked for more than 12 years in various capacities, from group leader, genetic discovery, and lead trade discovery, and up to program director. So he served there in various capacities. He used the conventional breeding to molecular approaches, including transgenics and genome editing technologies, to develop maize and other crops with important agronomically important traits. And later in 2000 till 2019 he was there, and recently he joined this NIPGR as a program director to lead the National Genomics and Genotyping Facility. So he has a lot of experience on working in the bench and leading the young scientists and motivating them and bringing products. So since I think he is the only industry person who is going to talk with you, who have developed the product, and these products are in the farmers field. Now I welcome Dr. Mohanty to deliver his lecture. So these are students from different parts of the country, from different universities. Some of them are started working in CRISPR technology. So I think your lecture will be motivating them for continuing there. Thank you so much.
Thank you. First of all, let me thank Dr. Viswanathan for inviting me for this particular occasion. And I'm always excited to be with the young guys. Hopefully, I can brighten up some of the things. Maybe you are thinking about the really gloomy scenario that's going on around. Let's see what technology has in offer for us. So what I would like to do here for this particular presentation is going to talk a little bit about CRISPR-Cas, and I'm pretty sure you have had enough experience. So I'll skip some of those slides, not get into dwell into detail, but give a couple of examples. I'll also focus quite a bit on the regulatory aspect, although I'm not going to talk about the regulation, but I'll give my thoughts. And also I'll discuss a little bit about the IP patent landscape, But first of all, I would like to start with a disclaimer that the views that I'm presenting today is my personal view. They are not from my previous or my current organization, okay? So let's start with the first slide, okay? As I said, what I'm going to talk about, the couple of applications in agriculture. There's some exciting stuff going on. I'll talk a little bit about the work that we did while I was working for DuPont Pioneer, and some of the new work that has been published, and then I have been part of the genome editing task force for DBT for last 5 years, and there is a lot of thought that we have put together what India should do, where we should go, and I'll share some of those perspectives with you. I'll talk a little bit about public acceptance and regulatory policy, followed by the IP landscape. So why you need advanced plant breeding? Because the farmers, they face real world challenges every day because of climate change, because of pest pathogens. So we need to provide them with climate smart seed so that they can provide food for us. And farmers have been doing creating new plants through breeding for more than 10,000 years. That's the history of agriculture. And CRISPR-Cas is a more targeted way to evolve healthy seeds. And this aligns very well with the SDG goals, for example, no poverty, zero hunger, good health, and things like that. I will skip this slide. I guess you have been hearing this for last 4-5 days, so you know what is CRISPR, so I will not get into it. But please remember the point that it started as a adaptive immunity in bacteria, right? And some of those early work happened in where? It happened in Danisco, which is a DuPont company, right? The first experimental evidence of this came from those fundamental work. This slide is important, so you may keep hearing from a lot of people, including some of our science leaders, a new technique, a new paper has come, like they'll say, oh, the old one is gone out of the window, this new. I can tell you, I'm probably one of the first one who started working on genome editing long time back. 2013 is the year that we started working on it. And I can tell you, even CRISPR-Cas9, which is one of the first one to come into picture, works really well. So for you, what you need to see is what works efficiently in your hand. Okay, and new technology came, base editor came. I don't want to jump into base editor. P editor came, so I want to jump into P editor. I don't think we should do that, right? It's always good and be open to newer technologies, but you need to see what works in your hand and work with that. As I said, this slide is important, but because in the basic version, what happens is very simple, right? So Cas9 enzyme, guided by the guide RNA, goes and does a double-stranded break, right? It does cleave the double-stranded DNA at the target location. After that, what happens is that these applications are based on that. So cellular mechanism, own mechanism is there for cell, they repair, right? And that there is no external factors involved in there. Depending on what kind of repair is happening, you have different applications, right? So for example, if there is a small deletion, because of non-homologous end joining, if there is a small indels or large deletion also is possible, that kind of process is called as SDN1. Similarly, as Dr. Bhatt said that when there are small changes, it's like SDN2, and larger one, for example, addition of gene SDN3. And the difference between SDN1 and 2 and 3 is, in this case there is template, in this case there is no template, right? That's a very simple difference to remember, that this happens you create double-stranded break, cellular mechanism takes over and does the repair. During the repair process, small deletions or large deletions or indels are happening, and in this case we are giving a template. Now, what are the challenges that we face for conventional breeding, right? So think about it, there is a plant which is drought tolerant, but this is of lower quality, and now I want to bring this higher quality trait, but this particular plant is sensitive to drought. So I want to bring this by conventional breeding. Even after so many generations of back process, you still have about 3 to 4% of the genome which is coming from the donor. Now please remember, recombination is not 100% that precisely it happens, right? So in a sense, what you may get is much more than that, right? And just think about it, even 4% of 5 GB genome is quite a bit of DNA that we are talking about, right? So that's a problem that a lot of times we have introgressed traits which are coming from wild progenitors, they have created problem of yield drags and things like that because they are not in the elite background. So CRISPR-Cas allows you to do that more precisely in a smaller scale, right? A smaller time frame, right? So in this particular case, so you have a line which is having a lower quality plant which is tolerant to drought, and you want to have higher quality plant which have got which is right now sensitive to drought, but you want to bring these two traits together. CRISPR-Cas does that, and I have an example later on to show you. For the gene editing process, we keep on focusing on the the real process part, right? But I would want you to focus on all these aspects. So it starts with guide RNA, transformation and regeneration, molecular analysis, as well as line development. Each of these steps are important. So what happens is that there is no unintended plasmid DNA in the final product. Outcome is possible Similarly possible through conventional breeding, and it is basically targeted mutagenesis. And I hope I'll be able to convince by my end of talk why I'm saying that. So as I mentioned that in my last slide, that it involves a lot of different steps. For example, you really need to understand high quality, like your DNA, right? Your genome, the target genome. You cannot be designing, like if you are working on rice, you are working with the indica rice, but you have designed your target from Nippon reference. What happens if you do that? One of the most important gene that we have found in recent years is SUB1A, right? SUB1 is what gives you tolerance to submergence, right? It's a beautiful gene that is not present in Nippon variant, right? There are several others, snorkel 1, 2, and there are multiple genes that I can talk about which are not present in reference genome. So if you design your target based on your reference genome, obviously you are going to get a lot of unintended effect, so then people talk about off target effects and things like that. If you understand your genome well, you have good informatics tool and infrastructure, these are not going to be the problem that you will face. Of course, once you have solved this problem, you need to have the ability to transform these to your elite genetics. You may have heard that not every species of rice is transformable, right? Even if you can do transformation, the transformation efficiency is really low, right? So this is also important criteria for successful genome editing. And last not but the least, you would like to see the tools with superior activity and targeting specificity. And this is not possible to, in the today's world to do everything in-house. So you need to have in-house expertise as well as use collaborators expertise. And in my previous organization, we did all this, so that's why we are quite ahead of the curve. So this is one particular example of Waxicon. You may have heard this from other authors probably as well. The USDA has said that it is not going to be regulated. So what happens in this particular case? This is a commercial corn, so which has got about 75% amylopectin and a quarter of amylose, right? In the Waxicon, we have a conventional product. When I say we, please remember my previous organization, right? So Pioneers right now Corteva has a product which is conventional Waxy, where you get more amylopectin. Okay, right? In this case, what happens is there is a time lag to integrate this, because this is a very old mutation in a different background. To bring this to elite, it's always slightly behind. So there is yield penalty, and because most likely because of yield linkage drag that I mentioned in one of my earlier slide, right? So the solution that thought about is that can you bring this Waxy mutation directly in our edit line, right? And that's what was done. This is the structure of the Waxy gene. Deletion was made between these sites, and a number of different edits were done, but this is one is represented here. So you can see this is the conventional Waxy, this is CRISPR Waxy, this is non-Waxy. So in this case, you can see the difference, and this can be found out by simple staining, right? The difference in color. So what has been done? So a mutation has been created, which is in our elite background, and the expected intended phenotype is there. No unintentionally integrated plasmid DNA, and like any product coming from this company, these lines are also going extensive field trials, right? Which is this picture is one of those trials, right, that is undergoing. So the difference and similarity. So the Waxy mutation is a 30 base pair deletion. The CRISPR one is full length gene deletion, but it gives the exact same phenotype. Let's look at the other example that we did in our lab in India. So we worked on this particular gene, bacterial leaf blight, and the way this is caused by Xanthomonas oryzae. It is one of the most important bacterial disease of rice. There are at least 39 resistance genes that has been cloned, okay, so far, and this is widely distributed across rice growing region. There is a strong race specific element, and there are many resistance gene has been deployed already. For example, XA21 is one of them, XA13, 5, 6, and XA33, XA38, and like. So what happens in this particular case? These pathogen, this bacteria secretes something called as TAL effectors, right? They go and bind to the promoter of the this particular native gene called as X13 or OSN3. It leads to elevated expression of the host gene, leading to this particular protein taking care of the copper T transporters from the xylem vessels, right, leading to disease susceptibility. So basically the bacteria through its stall effectors, they are able to hijack the plant machinery. What happens in resistance host, because there are small deletion, there are a number of different papers that you may have seen or may have come across, that if there are mutations or deletions or insertion in this particular 5' region of the particular gene in the promoter region, the bacterial Tal effectors are not able to bind and they are not able to upregulate the expression of the host gene. So there is basically copper accumulation in xylem vessels leading to disease resistance. Very simple mechanism. So what has been done? So basically the way you do it, you inoculate your seizures in this bacterial solution, clip the leaves, then incubate for few days, observe after 2 to 4 weeks, then measure the length. Very simple assay. So this is a particular example is the male parent and this is the female parent of one of our lead hybrid. So this is a positive line that has got X13 mutation already incorporated, and these are the variants, and you can see this is the control. Once you challenge this with Xanthomonas, you can see that really very strong disease symptom, whereas the edited line has got very little disease relation. Same way, these are a number of different variants were generated, and you can see in the variants which are edited, there are very few lesion length, whereas the control you can see how they are doing. And interestingly, I am not getting into the detail. These are I will not get into again the details. So these are different lengths of deletes. Okay, starting from one base pair to multiple base pair, and we see some really interesting phenotype, which I will not get into detail right now. There are other applications that are possible. So this is one particular example. In this particular case, from the transgenic work, we know that if you drive this particular gene, Argos gene, under GOS2 promoter, which is a moderately constitutive promoter, if you drive Argos protein, with this promoter, we know from multi-year field trial data that it leads to increase in yield under drought. There are publications there from DuPont Pioneer that you can refer to. So we thought that is it possible that we can create similar situation with genome editing. So what was done? There are two different versions that were created. So in the first version, GOS2 promoter was dropped in between the 5' UTR and the native GOS or the Argos promoter, right? That's one version. What we call as version 1. In the version 2, the Argos promoter itself was replaced with GOS2. Right. So these are two versions, but essentially they are similar, right. So what happens? So this is how the expression pattern of the native Argos gene, you can see very mild expression and extremely low expression, and look at the overexpression phenotype, right. In both these edited line, you can see, look at the axis, it's not a very strong overexpression, there are mild overexpression, and that's what you see. Right. So it's not a 35S kind of promoter or ubiquitin promoter, very mild overexpression. What happens in the field? This is again data worth 5-6 years in one table. You can see both the versions, version 1 and version 2, under drought condition leads to more than 5 bushel per acre yield. Okay, and this is under drought condition. Under optimal condition also about 2 and 3 bushel more yield. So we are able to recapitulate the experiment that we have done with transgenic lines with genome edited line as well. So this is another application. It has been shown from Dr. Viswanathan's former lab, Zhang Kangzhu's lab, that you can edit multiple genes simultaneously. This is a very elegant experiment that was done, and they demonstrated that editing 3 particular gene of this ABA receptor family, 1, 4, and 6. Leads to higher yield in the field conditions. Here is one more very exciting example. So Solanum pimpernifolium is a wild ancestor of tomato, which is Solanum lycopersicum, right? So this particular wild variety is wild species is a salt tolerant one, and also it can thrive under desert and tropics. Some accessions are also resistant to bacterial spot as well as tolerant to salt. So there are good qualities which are there, which is not there in cultivated tomato, in the wild species. But what are the challenges? The challenges is that in order to domesticate these wild species, you need to have a number of different traits changed. For example, plant architecture has to be different, flowering time and day length sensitivity has to be changed, fruit setting, fruit size, ripening, synchrony, nutritional content, all these needs to be changed, right? Now, this is how this particular one Pimpernellifolium LA1589 look like. You can see there are only two locule in this particular case, whereas the one that we eat will have multiple locule. You may have seen, like, if you cut a section of tomato, you will see. Now, what these particular group, there are three groups, Lemon et al., Shogun et al., and Lee, they did it. One of it is from Zach's lab in Cold Spring Harbor, one from China, and another from European lab. So what they did, all these three groups, they targeted some of the common gene, and some one of the gene was different. In this particular case, they tried to have this synchronized fruit ripening with SP, day length insensitivity by editing SP5G, enlarged fruit size either with clavata ocell or with FAS gene, which is in the next slide, and this is for the the vitamin C content or nutrition content. So what happened in this particular case, you can see this is there in the bar diagram. The edited line in this particular case, the Lee et al. Paper, they were able to generate about 140 independent lines. 45 of those lines are 4 genes edited simultaneously, right? And 68 lines are 3 gene edited. So what they found was that in many of these lines, which is listed here, at least 3 of them are here, you get, it's not a lot, right? But you can see between 15 and close to 15% of the lines have got multiple locules. Instead of 2 locules, now they have multiple locules. Now this is not domesticated yet, but you agree with me that there is improvement, right? Now let's look at the other example. In this particular case, the SPG, as I said that you want to use this domesticated one, they have to they change the architecture. If you edit this particular gene, this is how the plant look like, and you can see the sympodial index has decreased, so is the plant height, which is very clear from this particular one. Similarly, the editing of this particular facilitated gene, in this particular case, it also leads to change in flower structure. But now look at the wild type, tomato versus the edited one. They look pretty much similar to the cultivated one. So in this particular case, by editing multiple gene, they are able to increase the fruit size 3 times, fruit number 10 times. And by editing cyclic SICB1 gene, they are able to increase the lycopene content also by 5 times. So humans spend thousands of years domesticating something. This is probably 3 to 4 years lab work in the lab. So this is the promise of the technology, right? So if we are not able to use it, it is foolish of us that we are not able to take this exciting technology. That's what I wanted to impress on you, that this is the promise that is there. Now let's look into some natural phenomena. For example, if I tell you that all these 6 vegetables, they are the same biological species, would you agree? See, this is wild mustard, kohlrabi, kale, broccoli, Brussels sprout, cabbage, cauliflower. These are all coming from the same biological species. They something is modified stem, something is modified lateral bud, and breeders have selected these for certain traits, right? Let's look at the next example. These elongated or ovate tomato, how it has come into being. There is a 7 kb genomic fragment that has moved from one location in a genome to the other chromosome, leading to higher expression of this particular sun locus and leading to this particular phenotype. This happens in nature all the time. Okay. Similarly, between if you look into B73 and MO70, which are the two cultivars of maize, there are hundreds of copy number variation and presence absent variations, lots of indels and SNPs, right? There is a paper that this is the extrapolation of a paper, Arabidopsis paper. It says that if you grow that soybean in 1 hectare field, you can get upwards of 1.8 million novel SNPs in 1 hectare. This happens in the nature. So editing is happening in the nature all the time. So by trying to do editing in the lab, we are not trying to do something different than what happens already in nature. The point that I want to highlight here is that genome is not static. Genome is very, very plastic, right? So the changes are happening continuously. So similar example. So in this particular case, a NLB 18 sensitive line was made to NLB 18 resistant line by replacing the sensitive allele with a resistant allele. So this is, as far as I am concerned, this is no different than what is happening here, right? You are replacing in the same species. So the number of wide array of applications are there, for food and feed, industrial application, ornamental, and especially for vegetable crop, polyploid species where there is very limited diversity. This is a great way of bringing in diversity. Lot of examples that are being thought about potential solutions. This is one of the first product actually from genome editing. This is not done with CRISPR-Cas, this is done with TALEN, but TALEN is also genome editing technology, right? And this is coming from a small company called Calixt. So what they have done, they have reduced they have increased the oleic acid content. So it is 80% oleic acid, 20% less saturated fat, and 0 trans fat. So you can see in the commodity oil, the other percentage is quite high, whereas in the edited line, it is 80% of it is oleic acid, which is good quality, fat. Similarly, and this is commercial. This is being eaten in US. I have an example towards end. I'll show you. Similarly, this is low gluten wheat. As you know that there are people unfortunately suffer from this celiac disease, and this particular group has created targeted disruption in the alpha-gliadin gene, leading to up to 80% of immunoreactivity. So there are I am very excited about the prospect of CRISPR-Cas in the consumer traits. So far most of the focus has been input traits, but consumer trait is something that this technology has a lot to offer. So what are the advantages? I have personal experience of using earlier generation technology, and I can tell you that this is much simpler and much more efficient than anything that we have used earlier. It is a very precise editing tool, allows one to change DNA in a targeted way. This is much cheaper and faster than conventional transgenic technology. There are published papers, which shows that in order to deregulate one transgenic event globally, it takes up to $130 million. Okay, 130, that's close to 1,000 crore. Whereas there are paper which says that in order to bring a genome edited line to market, it may cost between $8 and $13 million. So basically, it allows even smaller institutes, smaller companies to play a role, right? For example, you see the first product is coming from Calixt, which is a relatively smaller company than the other bigger companies, right? As I showed you that it allows you the ability to do editing in the elite line. So the product development cycles are pretty fast, and hopefully I'm hoping for a long time. I don't know when it will happen. Eventually, hopefully, the regulatory cost is going to low, thereby allowing smaller players a level playing field. Even academic institution I think place like ICR, IRI can come up with products because the entry barrier is very low. Now, what India needs to do, I think as a country, India needs to have a holistic strategy. So you need to focus on capacity building as well as competition development, but we cannot ignore freedom to operate and IP issues, right? For example, I showed you that in the earlier slides, these three together only can make it really strong case. We have some excellent molecular biology and genomics lab, so the base is there. Capacity and competency development is essential. That's why you are here, because there has been a lot of investment coming both from ICR as well as DBT, training people, getting courses organized. As I mentioned, cost of development is going to be low, and I will elaborate a little bit more on the technology access and freedom to operate, because that is one area that may be is going to be a big challenge in the coming days. Let's talk about the public acceptance and regulatory policy. So what one expects is that appropriate science-based regulatory oversight, because the plants that will develop through CRISPR-Cas, can be developed through conventional plant breeding as well, and these and in most of the cases, gene edited plants are indistinguishable from the plant that could be produced with conventional breeding tools and which have a long history of safe use. And it is the characteristics of the plant, not the production method, that determines safety. And of course, protecting health and environment while promoting innovation, science-based policy, but it should accommodate new evidence and learning. What do I mean by new evidence and learning? I'll give you an example. Currently our regulatory scenario allows you only to use southern, right? If I do a single base pair edit, whether my southern is going to capture that? Probably I have to look for newer generation of technologies which can detect, right? That's what I want to talk here, new evidence and learning, right? We learn every day. Science is advancing at a very high speed. We need to be cognizant of that, and in our regulatory policy scenario, we need to make sure that we incorporate those advanced knowledge and learning. It should be cost effective and commensurate with the risk. Even if somebody from a small institute comes up with a product, you may not be able to sell that product because if the regulatory is burdensome, if people who has so much of money to get through the regulatory policies, right, and process. It should be transparent, consistently applied and enforced. And I think this point is also important, globally harmonized, because of the trade, it has to be consistent across the countries. And I think countries have started working towards that. But I want to share more about it from my personal perspective. So when you talk about regulatory policy, and please note, I have not put my title as regulatory policy and public acceptance. I have put public acceptance first, regulatory policy second, because that's the history of our country, right? Even the GM was accepted by the foreigners first, and then the policies came, right? So what I want to dwell upon here is really complex and nitty-gritties of lot of different aspects. For example, we tend to focus on the regulatory policy. Okay, that regulatory policy has to be this, that, right? But regulatory policy doesn't happen in isolation. It happens only when there is a willingness from the government, right? At the same time, there has to be public acceptance and social license for the technology, right? To me, this will happen only when we are meeting unmet needs and farmer welfare and consumer benefits are also taken care, right? If you can take care of all these, I think things are going to be green, right? If you are not, there are challenges. Having said that, in the lab, when you are doing technology development, please do not ignore intellectual property as well as freedom to operate, right? Whether you have freedom to operate with the stuff that you are working on. Can you really make a product and sell it? You need to be cognizant of that, and if not, then you have to find avenues, that how can you have that FTO? And this is what this is what decides the application development. So this needs to be looked into in a more holistic way than we tend to see this in isolation. So I am working in the lab, so I'll focus on technology development. Somebody is a specialist in regulatory policy, talks about regulatory policy. No, they are interlinked, and we have to see this holistically. So this is what I was talking about, this calyx oil. This is a party going on in a park, and they are eating a lot of stuff, including fries, which is which is using this particular oil. This is a healthier oil. This has got a lot of good characteristics, so people are happily eating in the United States. From the regulatory point of view, Australian government has very recently, a couple of months back, has stated that the SDN ones, they are not going to regulate. Okay, there is still discussion on SDN 2.3, and Dr. Watt spoke about some other countries. For example, Argentina. Argentina is one of the most progressive country. In fact, Argentina and few other Latin American countries are leading for the regulatory point of view. They have a very clearly defined path for success, which other countries doesn't have yet. I wanted to talk about the CRISPR-Cas. This is really challenging, and depending on if you are following carefully or not, you may be mistaken. So this is a very big debate going on between two big institutions, UC Berkeley as well as Broad MIT. And most people think about this is the story, okay, but that's not the story. The story is here. So they are not the only players, so Broad Institute and Harvard, along with New York Genome Center, Rockefeller, and University of Iowa, they are on this side, okay. And you see Berkeley with other, others, Caribou Biosciences, that's on the other side. But there are other players, for example, University of Vienna, DuPont Pioneer, Vilnius University, Emmanuel Carpenter. These are also important players, right? And each of them has their companies, which they have licensed technologies to. So you want to be operating at this area, and the landscape is pretty complex, right? But it's not that everything is lost, right? So let's look at this is a this is something that I have taken from a paper published in Nature Biotech last June, which talks about that what is happening about the worldwide CRISPR patent landscape. It says that private companies have filed 27 and 5% of these plant and farm animal patents, right? And you can see that most like private companies with most deposits are DuPont Pioneer, my previous company, which has got at that time 12 patent. KWS has got 5, Kgene has like 4, and Dow 3. Now Dow is part of DuPont, so this you can combine these two, and Deviant Technology China. So private companies have quite aggressively work in this area. The dynamics is changing. Okay, this is a title that came in Science. You may have seen this particular issue of Science. If not, I would strongly urge you to go through the articles. I am citing one particular article by John Cohen. What it says is very interesting. So for example, you can see the number of publication coming out of United States and China is very small difference. China has almost caught up. Look at the technology part, so this is almost equal. Whereas applied sciences as well as agriculture, China is much ahead of United States as per this article. Okay, and in some other areas as well, agriculture is there, application technology development. So China has caught up. Although they started late, they have caught up and they're marching ahead. That's why the slide that I talked about India, we really need to pull up our socks and make really good progress. So what is the way forward? So far people are very used to seeing this kind of funnel. The funnel is that you do discovery work here, do our research and development, and then some product gets developed, gets to the market. But world is changing. This is from Professor Henry Chesbrough. He's a professor at the Haas School in UC Berkeley. He is the one who first talked about this open innovation model. So now, as per this model, the funnel is much more wider. Now other players are coming, for example, consumers, the employee groups, experts, subject matter experts sitting outside can also contribute to the product development. And it doesn't have to be inside, it can be spin-off. So it has become much wider, right? So that means in simple words, there are smarter people also working outside, right? So what you need to do? You need to collaborate with people, right? So this is one particular example which I was also involved. So this is where ICRISAT signed a master alliance agreement with Corteva for access to CRISPR-Cas as well as plant transformation technology. So and there are multiple people from ICRISAT who has visited Corteva headquarters, got trained, and then they are doing work in India now. Other examples, so ERI very recently also signed agreement, to get that access to technology because they want to accelerate the development of improved rice varieties they got from both Corteva as well as Broad. This is the most latest one, as per this DuPont Pioneer and Broad, which is known as Corteva now. Broad Institute joined forces to enable democratic CRISPR licensing in agriculture. So I'll read it out. This is agreed on a joint non-exclusive licensing framework for agriculture use that continues to provide non-exclusive access to IP from Broad Institute, co-owned and co-owned with its collaborators, including Harvard University, MIT, New York Genome Center, New York University, Rockefeller, University of Iowa. I mentioned a little bit earlier, and provides non-exclusive access to foundational IP from Pioneer and to IP from the licenses that Pioneer gained through access through Caribou, ERS, and Vilnius University. However, please remember licensing limitations are there. So CRISPR-Cas technology for gene drive and tobacco products and for human use are not part of this one. If anybody wants to get license, these are not allowed. This is my last but one slide. So please remember, technologies are like, the world is changing. Technology is coming at a really breathtaking speed, and we need to integrate all this technology. For example, this is this is a nice article coming from Lee Hickey's group. This is how the traditional breeding happened. It takes a long time, because crossing line development and finally product development. Because of advances like double haploid, speed breeding, genomic selection and genomic selection with genome editing. Now this cycle can be reduced much smaller, right? This is the exciting possibility. So hopefully I was able to convince you this is nothing but precise breeding technology. It uses genetic material from the target plant only. Lot of variety of application, including on polyploid, as I mentioned earlier. Collaboration is the way forward, and rate of technology adoption is going to be directly proportional to the regulatory regime. So that's my belief. I would like to thank Neil, Tom, Jeff, and Masha from my previous organization, Corteva, for being kind enough to allow me to use some of them, slides and present some of the work that we did, and some of the work from the company which my colleagues in Johnston were doing. I would like to thank the group leaders in my previous organization, Dr. Venkatatava, Dr. Rita Gandhi, Dr. Amit Das, Dr. Bhojraj, Dr. Priyanka Vairi, Dr. Fanikant, who were actively involved in genome editing work, and also Sundar Reddy, Girish, Nilesh Sapkal, Janardhan Bodepulli, Dharmendra Patil, and Sam Sura, who worked on genome editing in the lab. So with that, I would like to thank you for your patience hearing, and would be happy to answer any questions. Thank you. No questions? Can I ask question? If you guys don't ask, then I'm going to ask question. So where is India from Dr. Bhatt's talk? Where is India in terms of regulation for genome edited products? Yeah. Diplomatic answer.
We have covered everything.
Right. So currently, unless there is exemption, see, there has been a lot of work that has been done. It's not public yet. Hopefully, it becomes public pretty soon. There has been a lot of work that has happened. But as of now, everything is covered under the 1989 Act. So what do you guys think about the potential? Actually, Dr. Viswanathan himself is doing a lot of good work here, so I'm pretty sure you have got experimental hand there. Okay? It can really take few years away from product development cycle, because you can get the traits much faster. All right, if there are no questions, thank you so much once again. Okay.
So on behalf of all the students and my own behalf, we are thank you so much for kindly coming and delivering this lecture. Thank you so much.
So we have to go there.
Thank you.
Thank you. Thank you.
Thank you.